Good morning, everyone. Today I am going to discuss classification of urochordate. In my earlier presentation, I told you that urochordata is a subphylum of phylum Chordata, and as the name indicates, uro means tail. That means notochord is present only in the tail region of the organisms belonging to this subphylum. Now, if you talk about classification of subphylum urochordata, let's first talk about the general characters. Then adults are generally sedentary. Sedentary means they are fixed to substratum, whereas larval form is motile. That's why we say that we have retrogressive metamorphosis in urochordate. This subphylum is also termed as tunicata because here the adult body is enclosed within a leathery test or tunic formed of a cellulose-like organic substance termed as tunicin. The notochord is present only in the tail of larva and it disappears in the adult. The dorsal tubular nerve cord is found in the larva and it is replaced by dorsal ganglia in the adult. Later on, when we will do type study of Hadmenia, which belongs to subphylum Urochordata, you will get the idea of what is dorsal ganglion. Basically, in urochordates, you don't have a well-developed brain and a spinal cord. Instead, you have a small structure which is termed as dorsal ganglia, which is basically the nervous tissue in urochordates. The pharynx is perforated by numerous apertures called stigmata formed by larval gill slits, and these stigmata basically gives a basket-like appearance to the pharynx. And why this basket-like appearance is required? Because all the members of subphylum Urochordata are filter feeders. That means they filter their food from the water. So in all the members belonging to subphylum Urochordata, an in current of water enters the body of the animal filters the food from the in current coming water with the help of pharynx, and then the water goes out of the body of animal. The larval tadpole is motile and undergoes retrogressive metamorphosis. That means it changes from a better developed larva to less developed adult. And how the larval form is better developed? Number one, larval form is motile. Number two, larval form has character of chordata. That means it has notochord, though only in tail region. So examples of this subphylum Urochordata are Hadmenia, which is termed as sea squid. Then we have Dololium, Salpa, and Botrytis. This is Hadmenia, which is sea squid, is in your syllabus, and we will be doing detailed study of this animal. Then if you talk about classification of subphylum Urochordata, then it is divided into three classes: Acidaceae, Thalassae, and Larvaceae. If you talk about class Acidaceae, it includes all the sedentary tunicates and the body is covered by test. Obviously, if it is a tunicate, its body has to be covered by test. Pharynx is large and contain gill slits. Notochord, nerve cord, and tail are absent in adult forms. These are bisexual animal and life history includes a typical tadpole larva. About classification of class Acidea, then it can be divided into two orders: order Enterogona and order Plurogona. In in order Enterogona, we have those Acidians which have one gonad in intestinal loop. Like in Plurogona, you have two gonads. That means gonads are paired. Along with it, in Enterogona, neural gland is ventral to the ganglia. I told you earlier also that ganglia, or which is also termed as dorsal ganglia, is basically a structure which is related to nervous system. Fine. And in Plurogona, neural gland is dorsal to the ganglia. If you talk about the examples of Enterogona, then we have Acidea and Siona, and in Plurogona we have Hadmenia and Botrytis. In this slide, you can see two pictures of each of the animal belonging to Enterogona and Plurogona. You can see Acidea. On the top you have the live specimen, whereas on the bottom you have diagram. Same is the case with Siona, Hadwinia and Botrytis. Botrytis is not, I think, visible to you, but you can always see it from the PPT which I will post on Google Classroom. Now if you talk about Siona and Hadwinia and Acidia, then you can see these siphons present. These siphons, finger-like projections which are extending from the body of the animal. Now these finger-like projections are basically siphons. As I have already told you that the members belonging to urochordata are filter feeder. Filter feeder means they filter out food from the incoming current of water. So these two siphons basically serve as mouth and anal opening. That means water enter from one of the siphon, enter into the body of the animal. The animal filters out the food and water exit out from the other siphon. Fine. Then class 2. Class 2 is Thalassia. Now, in this case, these urochordates are free swimming and pelagic forms. Now, what is pelagic? Pelagic means they neither live at the bottom of the water body nor they live at the surface. They stay in between in the water body, and their body is covered by transparent test. Now, this is important. Earlier, the members belonging to class Acidia has a translucent test, so you can't see the animal from the test. But here Thalassia, we have a transparent test. Branchial and atrial aperture are placed at anterior and posterior ends. I told you what are branchial and atrial aperture in the previous slide. Pharynx is small, gill slits number is less, nerve cord, notochord and tail is absent in the adult. Asexual reproduction is by budding, and there are bisexual animals. Tailed larva may be present or absent, and alternation of generation can be seen in life history. Now this is important. Alternation of generation means a haploid stage is followed by diploid stage, which is again followed by haploid stage. Fine. If you talk about the classification of class Thalassia, then it can be divided into three orders. Order Dolileolida, order Pyrosomida, and order Salpida. The first order, Dolilolida, is having a barrel-shaped organisms. Here you can see in the figure which is labeled as 5.8 here. You can see here a barrel-shaped structure is present, and barrel-shaped structure have clear muscle bands. You can see white stripes which are semicircular in nature. Then we have pharynx is small and number of gill slits will be small. Tailed larva is seen, and sexual blastozoids and asexual oozoid stage will alternate in life cycle. Then the second order is Pyrosomida. In this case, the order includes colonial forms. That means they live in colony. They are not solitary. Muscle bands are small and present at ends. Now in Dolilolida, you can see muscle bands throughout the body of the organism. Like in Pyrosomida, you have muscle bands only at the end, and gill slits are many. Tailed larval stage is absent, and the example is Pyrosoma. Then the last order is Salpida. Now this order includes organisms whose body is prism-like, and muscle bands are complete dorsally and incomplete ventrally. In Dololida, the muscle bands are complete both at the dorsal as well as ventral side, whereas in Salpida, muscle bands are incomplete. Muscle bands are incomplete dorsally, complete dorsally but incomplete ventrally. Only one pair of gill slits are present. Tailed larval stage is absent, and life history includes alternation of generation. Then, well, this is all about the classification of subphylum Urochordata, but the one thing you can do is you just type Urochordata in Google and see whatever information you get about this subphylum. You will come to know about various creatures, various organisms which are included in this subphylum.